Hello everyone. Today I will give a talk about Advancing Nuclear Medicine Imaging 2. Part 1. Gene Imaging. First, definition. Gene imaging is to use radionuclide labeled agents, we call it probe, to display the functional dynamic changes of gene and its expression products at the level of DNA, mRNA, or protein. 2. Classification. There are many methods of gene imaging. Here, I will only talk about the two main kinds. The first one is reporter gene imaging. Reporter gene imaging can be used to monitor endogenous gene imaging, study gene delivery and expression in animals, and to select the best gene delivery method. Because reporter gene imaging does not need to develop a specific protein probes for each new gene studied, it has become a general imaging method. The principles of reporter gene imaging. The reporter gene driven by a promoter can be transferred into the cell. If the reporter gene is expressed, the expressed protein will be captured by the probe targeting the reporter gene protein. If there is no protein encoded by the reporter gene in the cell, the probe enters the cell and falls out. Is quickly removed from the blood. The reporter gene can be expressed protein encoding the cell or the cell membrane surface protein. Different reporter genes can be studied by different promoters. For example, see this picture, micro-PET imaging of herpes simplex virus type 1 thymidine kinase, we call it as HSV1-TK in transgenic mice. In every cell of transgenic mice, there is a HSV1-TK reporter gene. However, because the reporter gene is driven by albumin promoter, it can only express protein in the cells with albumin expression, such as liver. See this picture, HSV-TK imaging. The micro-PET tomographic imaging shown was obtained about 60 minutes after the injection of fluorine 18 labeled FHBG. This is a probe specific for the HSV1-TK reporter gene product. Tumor treated with this virus expressing HSV-TK protein, so F18 FHBG targeting this product showed the tumor clear compared with tumor without virus treated. And the second kind of the gene imaging is antisense imaging. Antisense image is based on the principle of the antisense complementation to prepare the oligonucleotide chain, we call ASON, which complementary bind to the specific sequence of the target DNA or RNA. By in vivo hybridization, displaying the target sequence. In antisense imaging, the antisense oligonucleotide of the specific sequence labeled by a radionuclide. They can be used as antisense molecular probe, which can achieve specific binding with a corresponding mRNA fragments in vivo. PET or SPECT imaging will be used to reflect the transcription of target DNA. See this picture, the whole process of antisense imaging. First, we should find ASON, oligonucleotide chain. Label it with radioisotope. For example, IDN 131 or F18. And then we injected this probe into the mouse model. This model carries a tumor. And within the tumor and within the mouse, the antisense complementary binding to its targeting DNA. And this targeting DNA may be the overexpressed gene of the tumor. And then outside of the mouse model, we imagined it with PET or SPECT examination. Then we can find this overexpressed gene. Current method for in vivo gene imaging. First, mark protein include expressed intracellular or on the surface of cell membrane. Imaging method mainly on nuclear medicine, for example, PET-CT, SPECT-CT. Now, available receptors on transporter-mediated gene imaging as follows. First, carcinoembryonic antigen gene, we call the CEA. We can use radioiodine label. And second, dopamine type 2 receptor gene. For example, carbo11 rectopride, F18, FSAP, FESP imaging. And third, sodium iodine sympater gene. We can use radioisotope labeled. We call the radioiodine imaging. And the final, like other specific cell surface receptor gene, radioligand imaging. Okay, that's all for this part. Thank you.